Hello, I'm Tom Martin from the University of California, San Francisco, Helen Diller Family Comprehensive Cancer Center. And I'm joined today with Blood Cancers Today Editor-in-Chief, Dr. Sagar Lonial of the Winship Cancer Institute in Emory, Dr. Shambavi Richard of the Center of Excellence for Multiple Myeloma in the Icahn School of Medicine at Mount Sinai in New York, and Dr. Peter Voorhees of the Levine Cancer Institute in North Carolina. And this is our first roundtable of discussion on BCMA targeted CAR T cell therapy. I'm curious in terms of what your experiences have been, Peter and Shambavi, in terms of allogeneic CARs. Have you guys done any allogeneic CARs, or do you think there's a space, an ideal space for the allogeneic CAR T cells therapeutics?
So I don't have personal experience with the allogeneic CAR T cell products, but certainly in the relapsed refractory, more heavily pretreated patient population, I think that's the niche where they make the most sense. As Sagar pointed out, the starting product in our myeloma patients may be compromised in a number of different ways. And, I also think that these patients are very difficult, with regards to controlling their disease in the lead up to their apheresis, as well as during the period of manufacturing for the autologous CAR T cell therapy. So in the relapsed refractory scenario, having access to something that you can give right away has a lot of merit.
We've actually, we have the Allogene, the 715 trial at our institution. We've done a few of these patients. I completely agree with what Sagar was saying earlier in terms of, it's seeing those, first of all, quite tolerable, great vein-to-vein time, essentially off-the-shelf products. So it's really not even a vein-to-vein, but essentially we're able to turn around and give the drug very quickly after enrollment. So that's a pro. Pretty tolerable and able to handle the response rates are pretty comparable as well. But I think the issue is in the duration of response, and so still got to work on that. And the other big thing is with the anti-CD52, because that does increase the infection risk considerably. And we've seen one of our few patients that we have done on this trial who literally got hit with, a shingles and then a CMV. It was just like back-to-back viral infection. So I think that is, and this is well known from this anti-CD52 from, previous with the CLL and other disease entities. But I think these are some of the things to kind of pay attention to, because just to avoid to improve the persistence and to avoid graft rejection or the product rejection, we're having to intensify this lymphodepletion therapy, and that is not without its cost.
Yeah, completely agree. The more intense lymphodepletion certainly lines them up, unfortunately, for more infection. And this is being used in I would say, a more challenging patient population, even though all these patients are very challenging because they're multiply relapsed. But these are the ones, because it's off the shelf, these are the ones that potentially could have rapidly progressive disease. And now we're trying this therapy with them. And so the response rates may actually be lower with allo CARs just because of the patient population versus what we do with autologous CARs.